Serial dilution is this very simple, useful, basic method that we use to dilute a given sample, be it soil sample or it can be sewage sample or it can be any sort of sample. We are going to take these samples and we are going to serially dilute it so that we are going to reduce its microbial load. Okay, so if we don't dilute these samples by the process of serial dilution, we are going to face some problem while we are going to extract a single colony, as we are going to get a lawn culture in our petri plates. Okay, how we are going to serially dilute our sample. Okay, serial dilution, as you can see in the picture, you're going to take a set of test tubes. um, mm, For example, we are going to take soil sample. This sample can be anything, but now, for example, to explain it to you guys, I'm going to take soil sample. Okay. So in the tube one, we are going to take 10 ml of sterile distilled water, and to that we are going to add 1 gram of our soil sample that we have collected. Okay. So this is our reservoir. Okay. This is when it is soil. If we are going to take a liquid sample, as in sewage sample, we are going to take 9 ml of sterile distilled water, and to that we are going to add 1 ml of our sewage sample, that is a liquid sample. This is how the reservoir is prepared. Now, serially diluting this is, we take 1 ml of our sample from the reservoir, that is tube 1, and we add it into tube 2. Tube 2 contains 9 ml of sterile distilled water, and to this we are going to add 1 ml of our sample from the reservoir. This serial dilution method is going to be carried out throughout from tube 1 to tube 7 or 8 until you want your desired dilutions. Okay, why we are doing serial dilution, I have explained this before also, but I'm going to emphasize on the reason why we are going to do serial dilution. Okay, so let's take this very basic example where you're going to I'll enumerate the number of microorganisms. Okay, this is not how it works when you're going to work in the laboratory. This is not the number that you obtain when you're doing it in the laboratory. But for example, let's assume that in our reservoir there are going to be 100 microorganisms in a reservoir. Okay, so when we serially dilute from tube 1 to tube 2, the number of microorganisms that might have been transferred from one tube to the other can come down to a specific number, which can be 80 or 60. Okay, this is not an actual number, I'm just assuming so that you can understand. And as in when we go ahead with this dilutions, we up till we reach tube 8, the number of microorganisms in our test tubes can come down to a um, number that is as much as 10 to 5 or 3. This doesn't happen actually, this is just how I am explaining it to you guys. So what I want to say is the number of microorganisms, the microbial load that we are going to obtain is going to reduce with every serial dilution. Okay, so there is one thing that you have to remember when you're serially diluting a given sample. That is key. You're going to add one. If, for example, if eighth tube, as you see on the picture, eighth tube is the last tube that you're going to serially dilute. Um, we are going to add 1 ml of our sample from tube 7 to tube 8 and mix it thoroughly. And from tube 8, we are going to extract 1 ml and we are going to discard that 1 ml. The reason why we are doing this is so that we can maintain the overall um, concentration of the serial dilutions. Okay, this is one thing that we need to remember. Okay, next, we're going to go ahead with spread plate method. After serial dilution, the next basic method that we are going to go ahead is serial dilution. Sorry, spread plate method. Sorry, guys. Spread plate method, in which we are going to take sample tubes, that is tube 5, 6, 7, and 8. Why these tubes? Can anybody answer why we are going to take tube 5, 6, 7, and 8?
Because in these plates, the microbial loads are less compared to the other tubes.
Okay, right. Great. That is exactly the reason. As we are going to serially dilute, the number of microorganisms that go ahead in the further dilutions is going to reduce enormously. So in tubes 5 and above, the number of microbial load is lesser compared to the previous tubes. Okay, so in serial dilution, we are going to take 100 microliter of our solution from tube 5, and we are going to pipette it into a solidified nutrient agar in our Petri plate. Okay, 100 microliters. Now, using an L rod, we are going to evenly spread this sample throughout the Petri plate, and we are going to incubate these plates. Okay, this is done with tube 5, 6, 7, and 8. This not just 5 or 5 to 8. Any dilution you take, the last few tubes are the tubes that you're going to take up for dilution. Okay, this is how we are going to do serial dilution and plating. Okay, microbial culture methods. Okay, we are done with serial dilution. Now we are going to look into various methods that we are going to use for culturing our sample. Okay, it can be pour plate method, streak plate method, or spread plate method. Okay, first we are going to go through pour plate method. Okay, this method we are going to take a sterile Petri plate, and we are going to pipette into that 100 microliters of our sample directly onto the Petri plate. Then we are going to add moderately warm liquid nutrient agar onto the same Petri plate containing our sample. Now we are going to swirl our Petri plate so that our contents are mixed thoroughly, and we are going to incubate. Simple. Okay, this is pour plate method. Next is streak plate method. Okay, so streak plate method, there are three different ways of streak plate method. We can quadrant streaking, continuous streaking, and radiant streaking. Quadrant streaking, we are going to go through it later on. We are going to go ahead with that. Now we are going to look into continuous and radiant streaking. Okay, continuous streaking, what we do is we take a solidified agar plate, Petri plate, and to that we are going to, and also we are going to take a loop full of our culture. Okay, you can see a point A on your on the picture. From point A, we are going to zigzag go through the plate from point A up till middle of our Petri plate. Okay, only halfway through the plate. Now we are going to rotate the plate 180 degrees, and from point B, which is exactly opposite to point A, we're going to go with the same method, the same streaking, zigzag pattern from midway, from the point B till midway. Okay. By this method, we are evenly spreading our sample throughout our plate and giving enormous distribution so that individual samples, individual colonies are formed while we do all this streaking. Okay, same goes with radiant streaking. The first we take a loop full of our sample, and at point one that you can mark, that is marked on the picture, you're going to streak 2 to 3 times in continuous parallel motions. Okay, and then we are going to flame the loop. Okay, we are going to flame the loop. This is an important point. We are flaming the loop so that the amount of microorganisms that are present on the loop are going to get disinfected, or they are going to die. Okay, now we cool the loop, and we are going to radially move from point 1 towards the circumference of the Petri plate. Okay, these can go up in radial directions. That is why it is called radiant. Okay, radial motions up to 8 to 9 radial streaks can be done in one Petri plate. And after doing these radial streaks, we are going to again flame the loop. Okay, so we are going to kill the microorganisms that are there on our loop. Okay, now after cooling the loop, we are going to go horizontally through the streaks that we have done, the radial streaks, so that we are going to equally distribute and increase the chances of formation of individual colonies on our plates. Okay, this is how radiant streaking is done. Okay, next is spread plate method. As we had seen during serial dilution, this is what we are going to do. Is we are going to pipette out 100 microliters of our sample onto our Petri plate, and using a L rod that you can see, the L rod, we are going to evenly spread the sample throughout our Petri plate and incubate. Okay, after incubation, the next step is observing our sample. Like after incubation, usually the incubation ranges from 24 to 48 hours, and usually the temperature that is maintained for soil sample, if you consider that, it is 37 degrees Celsius. So we incubate our plates and then we go ahead with observing the colony morphology. Next is quadrant streaking. Quadrant streaking is this very commonly used basic technique in our laboratories to isolate simple, pure cultures from a mixed culture. Okay, this is like this most efficient method that we use in our laboratories um, to obtain pure cultures. Okay, so quadrant streaking, what we do is we take a loop, we take a loop full of our sample, and at the point marked 1, the area marked 1, we are going to continuously streak, okay, in sweeping motion throughout area 1. Okay, this should be done very delicately because you're going to do it on an agar plate, and little extra pressure with your loop onto the agar plate, it's going to stab the plate, and this is not a good sign. Okay, you're going to sweep on the area 1. Okay, now the plate, you have to rotate it 90 degrees, and we are going to flame the loop. Okay, why we are flaming the loop? Can anyone say why we are flaming the loop here?
Just
Sterilization.
Yeah, so we are killing the microorganisms that are present on our loop. Okay, great. um, Now we are going to go ahead with again the same sweeping motion on area 2. Okay, we are going to go ahead with second streaking, and we have to make sure that only one or two streaks are going to coincide with our previous streak. Okay, because our loop is sterile, it doesn't have any microorganisms. 1 to 2 streaks from our area 1 to area 2 is going to like pick up some 2 to 3 microorganism, and we are going to spread it in area 2. Okay, the same method again. Now turn the plate 90 degrees and flame the loop, and in area 3 that we have marked, we are going to go ahead with again streaking in sweeping motion. Okay, this is done. And fourth time, we are going to turn the plate 90 degrees, but we are not going to flame the loop. Okay, we are just going to go ahead with the fourth streaking, which is the tail, what we call. We streak the fourth in the fourth area, and we put the plate for incubation. Okay. Um, okay. One question I have for you guys is we have flamed the loop previous 3 steps we flamed the loop, but in the last method we did not. Can anybody explain why we didn't do that? Did you guys understand the question? Like, why do we not flame the loop while we are going to streak the tail streak?
like I don't know, it may be wrong also, but like whatever I understood, like previous also we have we have flamed just to reduce the number of colonies and to get the isolated one. So in the fourth, if we'll do that, I think getting the means desired colonies or isolated colonies, the chances of getting will reduce or something like that. I don't know, it may be wrong also.
No, no, you're right actually. If we flame the loop on the fourth time also, then the number of microorganisms that you're going to obtain in the fourth streak will be very reduced. It can be almost near zero. Okay. So that is why we are not going to flame the loop when you're going to go for the tail streaking. Okay, this is how quadrant streaking is done. Now we are done with plating our samples. Next is after incubation, we go ahead and check for the colony morphology. Okay, colony morphology is this basic first step towards our analysis. Ki what organism are we going to go ahead, or what organisms are we observing in our plates? Okay, so colony morphology is usually analyzed by colony form, which is the shape of the colony, colony elevation, that is how much it is raised from the media surface, and colony margin, which is the margin or the edge of the colony that we are going to observe. Okay, so colony form, we are going to view from the top. So we are going to go, we all we can see is circular colonies, which are circular and in uniform shape. One example for circular colony form is Staphylococcus aureus. You guys have heard of this name? Yeah. That is one example of circular colony form. Second one is irregular. Okay, irregular form where you are going to find uneven structures of the colonies. Okay, and one example is Streptococcus mitis. Okay, next is filamentous. Filamentous is, you see thread-like structures that are spreading throughout the edge of the colony. Okay, one basic example that you can find with filamentous form is Nocardia species. Okay, and colony forms that have rhizoid forms. This is, they have like root-like structures that are spreading throughout in the plate. Okay, and one example for this is Bacillus cereus. Okay, this is colony form. Next is colony elevation, which is the rise of the colony from the media level. Okay, this is going to, this we are going to view from our eye level. We are going to keep the petri plate at our eye level and see how elevated our colonies are on the petri plates. Okay, so this can be raised, which is slightly elevated on the media. Convex, which is dome shape. Flat is, there will be slight elevation, but it is not as much as you can see in any other type of colony elevations. It, there is not much elevation. This is flat. Okay, umbonate. Okay, this is one kind of elevation where you can see raised colony with the elevated convex shape in the middle, in the center area of the colony. Okay, and next is crateriform, in which there is raised elevation, there is an elevation, but there is a small depression at the center area of your colony. Clear? This is colony elevation. Next is colony margin. Colony margin and colony form are kind of interrelated, but in colony margin we are going to only look at the margin or the edge of the colony that we are going to study. Okay, this can be entire, which is evenly spreaded, or it has sharp defined edges, that is entire. Undulate is where you have wavy cavity like formations at the edge of the colony. Filiform is like thread like growth, as you can see in filamentous. The same way here, filiform is one kind of edge that you see with thread like growth. Okay, next is curled. Okay, this is where you find wave like layers spreading from center of the colony to the circumference, like towards the edge of the colony. Okay, these are curled margins. And lobate is where you find finger like bumps or branches that are extended from the colony. Okay, these are the various morphological features or morphological characters that we observe on a petri plate after incubation. Okay, this is, this ends our topic for today. This is what you're going to start learn for today. If you guys have any doubts, without hesitation, please go ahead and ask us. We are here to answer your questions. Anything related to this topic, we are here. We are going to solve all your doubts.
Hi. I'm Mohammed Shaibi. The informative session was actually delivered. It is very good with the detailed instruction. I do have a doubt on the topic that you covered at last on the colony morphology. Is this can be viewed under a microscope, the filamentous rhizoid, whatever you said, or the macroscopically in eyes itself, we can see those filamentous and rhizoid morphology?
Yeah,
This is
that's
not microscopic. This is
in the petri
macroscopic.
plate.
This you can see it in the Petri plates with naked eyes, not under microscope. This is directly when you look at the Petri plate, these structures are seen.
Okay, okay, okay. Yeah, thank you.
Okay. If anyone has any doubt, they can ask us. We can just sum up like whatever we taught you today. And I'm sorry if I was too fast. If you have any doubts regarding any media composition or anything, you can ask me again. And these are the topics we covered today. The first, we introduced you to microorganisms and what are those. And again, the basic apparatus which we use in the laboratory. And these are the basic ones which we use, which we come across every day in the laboratory and use them. Then we came to sterilization topics, which included sterilization by autoclaving and chemical sterilization, filtration, and other methods like dry heat, UV, and phlegm. Okay, and then this is at a glance where you can see all the sterilization is given. That you can do it by heat, filtration, chemicals, and radiation. Then we moved to culture media, where we discussed about how you can provide nutrition to the bacteria in a solid, in a liquid, and in a semi-solid media, and like the usage of all three. Then we talked about agar and why is it used. Also, always keep in mind it doesn't have any nutritional value. It just solidifies. It just helps to solidify the media. Okay. And then we came to agar slants, deep tubes, and plates. And I discussed like why do we need them and why do we use them. Then we came to classification of media. If you have any doubts in this, because I've seen my friends and juniors struggling with classification, because this is something which confuses us all the time. But I want to make sure that you understand that media can be selective and differential and enriched at the same time. Okay, like it's not like if a media is selective, it's only selective. It can be triple, like it can be clubbed together in a media, and we use them in everyday use. So like we call it selective, sometimes we call it differential, but it has multiple purpose all at once. So this is selective, where it inhibits some organisms but helps or supports the other, like the phenylethyl alcohol agar inhibits gram negative but supports gram positive. Then again, differential, where you can differentiate with your naked eyes that okay, this is my interest of bacteria, and okay, this is not, because the color of the media changes or the color of your colony changes. So it's very easy to identify that, like for example, in this, as I explained, pink is for E. coli and the other non-fermenters will give you yellow color in the plate. And in enriched media, these are generally used for fastidious organisms which have higher nutritional requirements. We don't generally use them, but yeah, for like other uses, we have to use enriched when the bacteria is fastidious. For example, streptococcus. And I also explained about gamma hemolysis, alpha and beta, and how you can identify that okay, this is gamma, this is alpha, and beta. Beta always has complete destruction of red blood cells and the use of hemoglobin. And I hope now you all also know that how hemoglobin is coming into the media. Because of the lysis of RBCs, it is coming into the media, and the bacteria can reduce the hemoglobin to methemoglobin, and the color changes. So in the first one, no lysis is taking place in the gamma, as you can see in the picture. No lysis is taking place, and hence the color is not changing. Only the bacteria is growing in it. But in alpha and beta, it's clearly changing. Then my friend discussed about serial dilution and why is it done to, of course, reduce the microbial load. As we all know that there are like n number of microbes we deal with every day. So we have to reduce the concentration before obtaining pure culture. Then she also talked about pour plate, streak plate, and spread plate methods, and why are different methods used for different, I mean, obtaining cultures of different types. And then quadrant, the streak plate method has quadrant, continuous, and radian streak. Generally, in laboratory, we go for continuous and quadrant. And the spread plate method, as you all know, that it is done by spreading the culture in the petri plate using a L rod. But make sure that your rod is very much sterile, or there are high chances of contamination here, because these we are not putting in autoclave. So make sure that you like dip it in alcohol or flame it properly before using it. And again, in the quadrant streaking, they explained it beautifully how you need to do it. And like you need to flame it again and again, flame your loop again until it's red hot. You have to do that and make sure that you don't get any contamination. And then after obtaining your culture, you have to check for the cultural characteristics, like the morphology, the colony morphology that we are checking. In the petri plate, you can see this, and you can say, you can check that if you, for example, are obtaining E. coli, you can check the colony morphology in Google, because we already have everything. And then you can check whatever you are getting and you can match them. You can understand if it's E. coli or something else. Then you can go for gram staining and other staining, which we'll be talking about tomorrow in our session, which will again start at 5 o'clock tomorrow. So I hope to see you all again tomorrow in our session. And now, again, if you have any doubts, any questions, even if it's a simple question, you can ask us. Don't hesitate to ask us. We always had this barrier between us and the professors that we were not able to ask them freely. But now we are here to help you. We will answer your questions. I think if you don't have any doubts, we can just wrap up today's session, and we will meet tomorrow again at 5 o'clock, and we'll be talking about different kind of stains and the staining techniques which we use in laboratory. Thank you so much for joining, everyone. Uh,
Thank you so much, everybody. Now you can leave the meeting. We're done for
yeah.
today. Thank you.